Thank you very much. So today I'll be talking about the triumph and tragedy of DNA evidence. You've all heard of DNA. It's this way of identifying people. There's other things as well. But there are problems with DNA, and in 2009, the National Academy of Sciences published a report on strengthening forensic science, and their question was, where's the science in forensic science? They were reasonably happy with DNA, but they weren't happy with the rest of it. So today I'll be talking about problems with DNA evidence as well, how it works and sometimes how it doesn't work. What's DNA biology? The human body is made of trillions of cells, and these cells, most of them have a nucleus, and our DNA is contained in that nucleus. We have two copies of the DNA in each cell, one from our mother, one from our father, and one copy is 1 billion letters of text written in a 4-letter chemical alphabet. And when you unravel it, it looks just like text. And one location of that text is called a locus. And we'll be talking about genetic loci. And the human genome is packaged up into 23 pairs of chromosomes because it's kind of a big encyclopedia. So there are 23 volumes for it. And the main type of paragraph that scientists look at for identification, and they've done this for about 20 years, is called a short tandem repeat. And the reason is there's a short word in the middle of the paragraph. This is in English. So here's a word root, it's 4 letters long, and it's repeated some number of times. A short word, tandemly repeated. What makes it useful is that different people have different lengths. Here there are 10 repeats of the word root. That's called an allele, is 10, the variant. But someone else may have 5 or 7 or 15, how many repeats there are. And so that gives variation that can be used to distinguish between people. So these paragraphs, about 100,000 of them in different chromosomes in the genome, they all start the same. This one is in English, take me out to the ball game, and it ends at the old ball game. But in the middle, the number of those repeats is different. And the number of repeats is called an allele or variant. And by measuring the length of the paragraph, scientists can figure out how many times that word's repeated. So if that paragraph were 4 letters longer, that would mean there'd be 1 extra word inserted. There'd be 11 repeats of the word root, and that allele would be 11. So you'll be seeing that the way scientists measure the alleles or the variants is by measuring the length of paragraphs. What we care about in identification is the genotype, the pair of alleles at one location that are inherited from a mother and a father, because we have 2 copies. They can be the same or different. This example, if we take a chromosome and zoom in at one location, there may be 10 repeats for the mother allele, 12 for the father allele, and that pair of alleles is the genotype 1012. And what scientists do is they measure maybe 15 different locations across your genetic encyclopedia looking for that pair of alleles. 1012 at one location, 513 at the next location. And because there are about 1 trillion possible genotypes, that's a 1 followed by 24 zeros, and not even 10 billion people on Earth, which is a 1 followed by 10 zeros, there are a lot more of these genotypes than there are people, so they can be used to identify. Okay? That's what genotypes are all about. The old sort of DNA interpretation that you might see on television is there's a large pool of blood at a crime scene. Somebody scoops it up, they analyze it, there's DNA from one person, and then this is very straightforward. The lab takes that DNA, they extract out the DNA molecules, amplify it to make millions of copies that can be detected, and then they separate it by size to find the alleles, and with a laser measure how much DNA is present with fluorescence. And they get a signal. With simple DNA, of all the possibilities that could exist, a 5 and a 12 as a pair of alleles, or a 7 and a 7, or a 9 and a 15, the data are so simple that you'll see just 1 or 2 peaks, to many of them the same or different. Here's the size, the number of alleles, 8, 9, 10, 11, 12, 13, 14. And the height is how much signal the laser measured. And if you see 2 peaks that are tall for a 10 and a 12, that eliminates the other 100 possibilities of this one location. And that leaves only one possibility, at 10 and 12. So simple DNA is really simple. The data pretty much tell you what the genotype is at every location. 2 peaks, 10 and 12. Persons at 10, 12. Later on, you can make comparisons. Most DNA evidence is not simple. It's not a cheek swab. It's not a pool of blood on a floor in Los Angeles, right? It's a mixture. Sexual assaults are inherently mixtures. A victim, an assailant, maybe a consensual partner. You can have 3 people in there, 4 people. Touch DNA from a surface. Maybe 10 people have touched this podium recently, of which the signals of 3 or 4 of them would be detectable. Most DNA is mixtures when it's actual evidence. And because it's mixtures of DNA from multiple people, there are a lot of different genotypes in there, and it's harder for people to interpret. This is the main method used by crime labs to interpret DNA evidence. We'll see what some of these signals look like, but there's a complex signal. It's not just the Twin Towers, right? It's a complex skyline. There's taller allele peaks. There are shorter peaks. There's just lots of different peaks. We'll see what computers can do, but what people do is they begin by simplifying the data. Instead of looking at the data that they see in front of them, they draw a line called a threshold. Anything over that line is real. Anything under that line, they determine to not be real. A very simple procedure. The result is this is the Pittsburgh skyline. We've got a bunch of buildings of the same height and a bunch of vacant lots. That's what the data is reduced to, which, as you can imagine, loses a lot of information. I mean, that could be Chicago, right? Reduced to buildings of the same height and vacant lots. It's not telling us about the evidence very much. So that's thresholds. And when they simplify the data in that way and they have thresholds, it gets very easy. They can look at the frequency of occurrence, how much each of these lengths or alleles occur in the population. One may be 10%, 5%, another one of these lengths or alleles might be 15%. And they just determine which peaks are at least at the threshold. They take the frequencies, add them up, square it, take 1 over the number, that's the match statistic. I mean, a grade schooler can do that on the back of an envelope. It's a very simple procedure. And that's why it's been popular for 20 years. It's simple. You draw a line, you add up some numbers and do some arithmetic, and out pops a statistic. At one locus, it's called the probability of inclusion. If you're going to multiply these numbers together from independent locations, say 15 different locations, that's called a combined probability of inclusion, or CPI. And CPI has been the subject of much discussion for the last 15 years in forensics. And I'll tell you what the problems are. So imagine most of the DNA statistics in the world have been done using CPI. Hundreds of thousands, probably 100,000 every year, maybe millions over the last 15 years. Most DNA evidence is mixtures. This is the most popular match statistic. One would hope that it's reliable, right? Wouldn't you hope that it produces something that tells you something? Since people are executed on it, they're freed from prison, put into jail, let out of jail, people's lives and freedom and protecting society from crime depends on accurate match statistics. How accurate is CPI? This is a study done by the federal government, the old Bureau of Standards, renamed NIST, National Institute of Standards and Technology. They did an interesting study. They sent out about the same simple 2-person mixture. So 2 people are combined, they're in the data, sent it out to about 70 laboratories. Most laboratories, crime labs in the US, most laboratories said, inconclusive, we can't say anything at all. But about 1/3 of the labs did put a number to it. Same data, many labs said that the number was in the tens of thousands. That was the smallest number. 4 zeros. 10,000 is a 1 followed by 4 zeros. There's a complete range of numbers. The biggest number was in the hundreds of trillions, a 1 followed by 14 zeros. So this was all over the map. People either said there was no information, or when they put a number to it, it ranged from tens of thousands that a jury would hear to hundreds of trillions. So certainly this method wasn't reproducible between different crime labs. Another problem that people published about 5 to 10 years ago was the notion of bias. You would think that if you have data, you take that data, you put it into a computer, it does some analysis, and you read the number. But that isn't how it works in crime labs. Instead, first, people choose their data. What's a peak? What isn't a peak? Which locations will I use? What won't I use? What exceptions will I apply for different thresholds? Is it really there or not? Then they look at the defendant, could be someone else, is usually a defendant or suspect, and then they decide, is the person in there or not? This is before any math is done. First, they choose their data, then they look at the genetic genotype of the person they're comparing with. If they decide the person's in there, which can mean guilt in many cases, or not in there, but if they decide they're in, then they calculate a statistic. This is not an unbiased process. In most of science, you don't choose which data to use. You don't look at the answer before you decide whether or not to use a calculator. That's the state of the art in crime labs. It's a very subjective process, and papers have shown that when people have reanalyzed without context, they can get the exact opposite answer, just by knowing what the answer is supposed to be, like who the defendant is. There was another study done in 2013 by NIST again. It kept growing. This is a snapshot where 100 labs responded. This was a 3-person mixture, and they asked if one of the people was included. Now, they knew the person was not in the evidence. He actually wasn't in the DNA. 70% of laboratories in the study said, he's there. And they gave a CPI match statistic of the type that would be used in court. Their numbers ranged from 9 to 340,000. So that's the state of the art. It doesn't work. Mixtures analysis just give no answers or wrong answers. In this study, 25 labs roughly said it was inconclusive, and about 5% of the labs got the right answer and said the guy wasn't in there, and some of them used computers. This began being noticed in criminal cases about 2 or 3 years ago. The District of Columbia crime lab was shut down, all the senior staff was fired, and then the FBI came in to do their version of CPI, which has 2 thresholds, not 1, which also doesn't work. That was the study. In Texas, they have had problems with labs. Many labs have been shut down, some of them reopened, and about 25,000 lab tests or cases were affected. And that's just where people are looking. The problem is the same in all crime labs across the US. A year and a half ago, I wrote a paper. Many people have written about this. This one was based on data from a crime lab. And I tried to answer the question, why is it that when you have 13 locations, 13 of these loci, which is what the standard has been in the US for the last 10 years, why do you always get the same number around 1 million? Whatever lab you're in, you always get the same number. You would think that different degrees of mixtures have different amounts of information, and many studies have shown that. When you really look at information, you can move from no information to a number like 1 trillion trillion, which shows the extent to which somebody is present in evidence, or you can go the other way. You can show the degree of exclusion, get a number like 1 in 1 trillion trillion, which is very small and excludes someone, and everything in between. Why were the crime labs always getting the same number? The answer is, it turns out that the CPI method's a random number generator. Every 2 tests you do, you get another factor of 10. You do 2 tests, the number's around 10. You do 4 tests, you get a number around 100. You do 12 tests, each 2 of those gives another 0, you get 6 zeros, you get a number around 1 million. So the main statistic that's been used is just a random number. Whatever the number of tests is where a person has decided that somebody's there, every 2 of those tests gives another factor of 10. And that's what most labs do.